to do. Yeah. So collaboration is really important to do the advanced research on utilization of organism-wide data for optimal cell annotation and gene regulatory networks, also on improved annotation of pan-genome reference assemblies with single-cell data and comparative analysis of single-cell data across species. So to achieve this, we need to have data and metadata standards and vocabularies and consistent analysis pipelines, which are really crucial for data sharing, annotation, curation, and integration. However, currently, most of the data and metadata is manually curated, and user-provided data metadata is not fair, and there's a requirement of best practice training, and there's no centralized annotation portal that exists. So because there's no portal exists to provide access to all public single-cell RNA-seq for agriculture species, so very little metadata is available for most such data sets. So data reuse is very difficult. So agriculture research output, quantity, quality, and reproducibility could be significantly improved if such community-wide resources are developed. So therefore, plant and animal single-cell researchers have been discussing some means to develop such global resources. So global repositories and tools to share data, metadata, and results that can facilitate community annotation of cell types. And also single-cell data sets can be used, applied to biology and breeding phenotypes, genotype to phenotype and genetic improvement. To facilitate this, Ag Biodata Consortium has also a single-cell biocuration working group. This group has several members from different organizations, Plant Cell Atlas, FANG, like functional annotation of animal genomes, and AG2PI. And main goal of this working group is to address the annotation challenges of single-cell RNA-seq metadata in plants and animals and establish a set of recommendations for member databases on management of single-cell RNA-seq datasets. So main goal is to also to facilitate in creating a cohesive community that will provide standards, resources, tools, and standardized single-cell RNA-seq datasets for fair data meta-analysis that leads to initial cell type transcriptome descriptions. So basically, we will review then best practice recommendations for integration of member database data with larger efforts such as public international sequence nucleotide databases, INST archives like GEO, ENA, DDBJ, Plant Cell Atlas, and Single Cell Expression Atlas, FANG, etc. So now I will not take that much time. I will go directly to the speakers. And I would like to introduce our first speaker, Dr. Sylvie Fexova from European Bioinformatics Institute, Kingston, UK. She did her PhD at Dr. Sarah Bray's lab in Cambridge, UK, where she investigated the role of Notch signaling in cell apoptosis and survival in Drosophila melanogaster. She joined Flybase as a curator after that, and then she moved to EMBL EBI. Now she currently works as a senior scientific curator in the gene expression team, where she looks after the content of the 3 resources the team maintains: the Array Express Archive, the Expression Atlas, and the Single Cell Expression Atlas knowledge base. She leads the Array Express Functional Genomics Submission Project and the maintenance and further development of its submission tool, Annotea. So now Sylvie is going to talk about single-cell data ingestion and metadata through Annotea today. Thanks, Sylvie. Please go ahead.
Right.
I'll stop sharing.
Thanks very much, Sunita, for the introduction and for the opportunity to introduce some of our work here today. Let me just start my presentation. So my name is Sylvie, and I'm the submissions project lead in the functional genomics team here at EBI. Our team currently maintains several resources for data submission and archival, which I'm going to talk about today. That's the repository for functional genomics data called RxPress and its submission tool Annotea. But we also maintain 2 knowledge bases, Expression Atlas and Single Cell Expression Atlas, where we reanalyze and integrate gene profiling data across species and conditions, and then show the results to share the results with the community via the web portals. Right. But today I'm going to focus on the submission tool Annotaire. What is Annotaire and ArrayExpress? So Annotaire is is a submission tool, and ArrayExpress is the archive, or at the moment it's basically a collection under the Biostudies archive. So the submission tool Annotaire says for our users to sort of put together their submissions, make sure that all the necessary bits and pieces are there. It has a temporary file holding storage for your data files. And when you're ready to submit, it will go through further processing and loading, and then it will be permanently securely archived in the ArrayExpress collection. And we also brokered the raw data file in ENA, which makes them accessible through SRA and Geo as well. So why we developed Annotaire? So the idea was to promote reproducible research and help users make their data submissions fair, and also make it a bit easier to create submissions which are part of journal sort of journal acceptance, so publishing requirements. So the what Annotaire does is that it provides templates that are configured to each experiment and sample type, and it then helps users to capture all the data related to an experiment, including protocols, background information, and so on. It ensures, or at least try to ensure high quality metadata by enforcing several data standards for microarray, high throughput sequencing, and single cell sequencing. It helps users to define the unique links between samples and the associated files. It also provides instant metadata validation feedback, and helps with quick and it's basically a gateway for quick automated accessioning when everything is fine. So Annotaire is a submission tool that can be used for any functional genomics data, both microarray based and sequencing based, and which includes single cell sequencing and sequencing based spatial methods. So basically, we can accept any data that come out of a microarray scanner or a high throughput sequencer. So it is basically what we hope a user-friendly web form tool to help collect all the data and metadata about an experiment. So this includes the background information about the experiment and the performers, information about the samples, protocols, including wet lab protocols and any post-analysis, and collect data files, which include raw data files that are mandatory, but it is encouraged to also submit processed data files, and you can do all of this easily through Annotaire. It helps with reproducibility of data and findability of data by adopting standards, as I mentioned, and it also adopts ontologies under the hood. So it will sort of prompt users to use ontology terms where suitable, but if none is available, you can, of course, enter any sort of free-form annotations. There's a mix and match system of templates according to the technology and sample type. And of course, we continue to develop these templates in line with advancements in technologies. So today I'm going to focus on the single cell sequencing. So the single cell sequencing template is the latest one that we've introduced, although it's been some years ago. And with sort of every template development, it's a bit of a journey. So we try to keep pace with the advancing technologies, and as a new group of technologies start to emerge, we try to accommodate it. But obviously, there will always be a bit of a lag, because at first these technologies need to get established, and also they the data standards for them need to be agreed. So we basically need to work with the community to agree what information is important to collect about a certain type of experiment. For the single cell sequencing template that we've recently, not that recently, that we added last, these are basically, it's based on this joint proposal of basically minimum data standards for single cell experiments, which is a joint, and here's a link to the paper, which is a joint work of our TD, ACA, HubMap, and other teams. And in Anotaire, this basically is reflected by additional sort of tab or a new tab with the single cell sequencing template, which captures the information that's specific for this technology, such as single cell isolation, single cell well quality, if it's a well-based experiment, library construction detail, and position and size of any barcodes, unique molecular identifiers, generally the content and identity of the individual reads, and any sort of post-analysis single cell quality data can also be captured using this template. So now I'm just going to briefly introduce Anotaire, the submission tool. So this is the landing page for Anotaire, and this is the URL. First, you need to create an account, and after you create an account, you can start using Anotaire and create submissions. I would encourage anyone new to Anotaire to have a look at our submission guide, which will sort of give you some pointers about what type of files are accepted, in which formats, what are the requirements. It does also give you information about excuse me, the various upload routes, any sort of time-saving tricks and features that Anotaire has to make the submission process easier, and also explains about what basically happens after submissions, how to make edits if you need to. So once you have your account and you log in, this is the page that you will see. There will be, if you're a returning user, there will be a list of all your previous submissions showing their current sort of status. And to start a new submission, you just need to click on the create button, and you will be presented with this sort of decision tree, as it were, where you can select your technology template, as well as the material template based on the type on the type of your samples. The decision on the sort of templates is an important one because it basically changes what type of attributes will be pre-selected and what type of information you will be asked to supply. And this basically cannot be changed later. You can customize and modify your template further, but if you pick the wrong one, you just have to start a new submission or write to us, and then we can sort it out for you. The sort of next step will ask you for a number of samples. You can also use the experiment design to further customize your template. But you can also just skip it and customize it later. But what the selection of experiment design does is that it will add certain type of attributes to your sort of to your template, and we'll select some of them automatically, pre-select those as experimental variables. So if you select a genotype design, then the template will assume that genotype will be an experimental variable, and it will be sort of pre-selected as such. So this, after you sort of make these initial steps, you will be presented with a page that looks like this, and this is your submission page. On the right-hand side, it's the upload file pane. And on the left-hand side, or the left half of the screen, you see basically the different tabs and different types of tables that you will that you then sort of go through and fill out all the metadata that are required for this type of experiment. Generally, we suggest people start with the file upload, because if you are uploading a lot of files or they're quite big, it can take a while. So while the upload is ongoing, you can use that time to fill out the rest of the submission form. There are several ways of uploading files to Anotaire. Small files, like smaller than 1 GB, can be uploaded directly through the drag and drop using the upload file button. But anything bigger would be too error prone for this type of transfer. And at the moment, you can use FTP or Aspera upload. And we also provide Globus upload route. At the moment, it's upon request, but we are working hard on including this, integrating this into Anotaire as an additional upload option. So once you decide on the way you wish to upload files or what type of files you want to upload, there's a detailed, you can just follow the instructions here. There's a sort of detailed guide to how to use the FTP or Aspera server. That can be Aspera upload route. So there's a detailed guide under this link, and this is basically the unique sort of FTP, so the sort of directory or space for your files for your submission. So this is the sort of the, folder name that you will need when uploading your files. And the last step is then to register your files into Anotaire by calculating the MD5 hashes, which is something like a digital sort of fingerprint of each individual file and their file name, and this will import them into Anotaire and allow you to start assigning. So so while your files are uploading, you can continue with the rest of the submission. And the next step, apart from filling out the general information or the contacts and any details of publications, would be to create your samples and add attributes to them, depending on the template that you've chosen at the beginning, and or any experiment designs that you've selected. There will be attributes sort of pre-selected for you, but you can also always customize this further by either removing, although some attributes are mandatory and basically cannot be removed, although you can not fill them out. But you can add more or remove some of them using this menu here. And if none of them are suitable, you can always create an entirely new attribute that would sort of better describe what your samples. So as I've mentioned at the beginning, Anotaire incorporates ontologies under the hood. There are several ontologies that are kind of running in the background, and whenever you start typing into your web form, into the attributes, it will try to offer some ontology term suggestions. But you can, if they're not useful, you can ignore them everywhere apart from the organism. That one has to be mapped to an ontology. To speed things up, you can always just you can also use this fill down value button that basically propagates whatever you've selected in the cell that you're currently in to all the cells below. So that's one way of speeding things up. We are trying to make the process as easy as possible by also offering a lot of sort of drop-down menus and automatic pre-fills. So for instance, when you're asked to fill out the single cell library information, you can select your technology from this list. If it's not there, then you will have to select other and then like manually fill out all the information. But if your technology is there, you can just select it and it will automatically pre-fill the rest of that form for you using the standard values. So you can modify this, of course, if you've modified the protocol a bit, but if you just follow the sort of standard manufacturer protocol, these should be correct, and that's another way to sort of speed up the annotation process for our users. And again, you can of course use the filled out value to then like propagate this to all the remaining samples as well. Annotator will also ask you to supply protocols. There's for each template type and technology type, there's a set of minimum protocols that have to be filled out. And you can add more depending on the particularities of your experiment by selecting from the menu here. Right. So this is the bit that I mentioned at the beginning. So while you're sort of filling out your metadata, your sample and experiment metadata, the files are being sort of uploaded in the background, and now if they've been all successfully uploaded, it's time to register them in Annotator. This step is important because it constitutes the first step in the file validation. So basically, we ask for the MD5 hashes to ensure that the copy of the file that we received is the same as the one that you have on your local copy. So basically, we then calculate the MD5 sums on our end, compare the values to whatever the users have provided, and if they do not match, that sort of suggests that the files have been broken during transfer and need to be refloated, and there's no point in running them through the next validation steps. So that's why we ask for this. It's not just to make life more difficult for for our users. But so once you have all your files uploaded and registered in Annotator, they will all appear here in the file upload pane, and we'll have the status uploaded. Once they have the status uploaded, they can be assigned to your samples. There's several ways of assigning files to samples. You can either just select them one by one from the drop-down menu, or if you have a lot of files, this might take a while and might not be very convenient. There is another way using the paste into column function. So if you have a spreadsheet with the file to samples assignment locally on your local machine, you can basically copy them in column by column and insert them into the table in Annotator. The only but crucially important thing, of course, is to make sure that the order of the samples in your spreadsheet on your computer is the same as the one in Annotator. Otherwise, this would misassign your samples. So another sort of functionality that I think can be is quite nice and probably and helpful to users is that at any point when you're doing your when you're working on your submission, you can validate it locally. This is a validation for against basically the standards and the metadata quality. So when you think you've finished everything, or you just click on the validate, and it will it will show you any warnings or errors. So anything that you might have forgotten, any required fields that have been left out, unfilled, will appear as errors. Warnings are suggestions. Errors are prohibitive. So you will not be able to submit if there are any errors appearing. So those need to be resolved. And there's and the validation result down here will list all the errors that have been identified and where and how to fix them. So I've prepared a short video because I don't think we would have a scope for a live demo. Just to show you the process as a whole. This is run at 2.5 speed. So no matter how well organized you are, you're probably not going to finish your submission in 2.5 minutes. But this basically goes through all the steps that you will need to complete to finish your submission. So here you can customize the attributes, you can add units to them. This is basically how you can fill out your sample attribute table using or not using the ontology suggestions. And then and then we sort of move on to the technical information about the sequencing libraries. This is all drop-down menus, and you can always use the fill down button to propagate the values to your samples. Then the next step is basically to insert all the at least all the required mandatory protocols, and then assigning your files. If you have a small number of files, like in this case, it's probably finer, maybe quicker to assign them individually. But there is the option to bulk assign files. And before you submit, you need to validate. When you click the submit to ArrayExpress button, the validation will run again. It will point out any missing fields, any omissions that need to be corrected. Some protocols, well, just the sequencing one, require information about the performer and hardware, but Anotair will prompt you for this, and that's it. Basically, that's how the submission works. After you, sorry. Okay. So once you've completed your submission in Anotair, that basically sends it to further processing and validation. So what happens next? You as the user and the researcher, you sort of complete your submission in Anotair using the templates. If it's a sequencing submission, it will go through FASTQ validation or raw data validation pipeline. You can submit your sequencing, your raw data sequencing file as either FASTQ or PUM files. It will then go through validation to ensure that the FASTQ files are complete and valid, basically. Once this is passed, and a stable accession will automatically be granted, and this will not change. You will be notified by email when this happens, and you can basically already use that submission in your manuscript, although further processing is required before the submission is finally loaded to the ArrayExpress archive, and the raw data files are also brokered to ENA. The whole process from submission to loading usually takes less than a week. And accessions are normally granted within 24 hours. Depends on how busy the pipeline is, how many files you have, how big they are. For microarray submissions, it's almost instantaneous because there's the validation there is much quicker. But for sequencing experiments, the FASTQ files have to be checked, or the raw data files have to be checked, which can take some time. And only after this is passed, the accession will be granted. But it is normally quite quick and should never be much longer than 24 hours. So once this whole process is completed, this is basically the end result, the final product. Your experiment will be loaded to the ArrayExpress collection in biostudies. And this is just a screenshot of that sort of collection, where you can search for particular keywords or filter by type, presence of different type of files. There are many filtering options on the right hand side. This is just a few of them. Pardon. And this is the experiment page itself, with its sort of stable accession, which has this format EM tab and a number highlighted in here. And it basically contains all the information that you've submitted through Annotaire. There are all these like subsections that you can open up to find the details. And there's a link to the corresponding records in ENA, and you can use the sample information and links data table view for an overview of all the experiments, the individual samples. So it just it lists the samples, all the metadata and sample attributes that we've got for them. A contracted sort of information, technical information, and links to the individual links to files for the individual samples. And yes, that's the end of my presentation. I just wanted to acknowledge the actual genomics teams who we have a recently new team leader, Christina Adams, and our team basically consists of 3 sub-teams, and it's the Annotaire and ArrayExpress sort of setup is maintained by this sub-team. So, and I'll be happy to answer any questions. And thank you for your attention.
Thanks, Sylvie. Thanks for the wonderful talk. I don't see any questions right now, but maybe later on. Let us move to the second speaker right now. And hopefully, I would like to request participants to ask questions. And please feel free to ask questions at the QA space. And then we will take all the questions at the end of second talk. Okay, so I would like to introduce Felix now. Felix did his PhD in signal processing and computer graphics from University of East Anglia. Currently, Felix works as a COPO platform leader at the Phenome Initiative. COPO is collaborative openomics. It is a cutting-edge platform developed to facilitate the management, brokering, and validation of bioinformatics data. Felix is very passionate about the practical implementation of the FAIR principles in research data management. In his guidance, COPO has evolved from its focus on genomic data to become a versatile tool for metadata standardization and integration. As the sole validator for prominent projects like the Darwin Tree of Life and European Reference Genome Atlas, COPO has successfully proctored over 60,000 samples, positioning itself at the forefront of genomic research data management. And now let us hear more on COPO directly from Felix, how COPO is used for submitting data and metadata to ENA. Thanks, Felix, and we are excited to hear from you. Go ahead.
Thanks, Anita. Can you see my screen okay?
Yes, we do.
Good. Okay. So thanks for inviting me to talk here. As the introduction said, I'm the lead developer for the COPO platform, which is a mature brokering service for data and metadata in the biosciences. COPO stands for Collaborative Open Omics. I'm a research software engineer as well and a data scientist working in Irene's new group here at the Earlham Institute in the UK, not far from EBI, in fact. And I'm going to talk to you about COPO and that I've been doing. Okay, so there we go. So I always start these talks with a quick slide on what FAIR actually means. So FAIR is an acronym that stands for findable, accessible, interoperable, and reusable. Sorry if I'm teaching people to suck eggs, but I just want to make sure we're all on the same page. So assuming you have a study, this study contains some data. Now, to make this study fair, the first thing we should be doing is putting it into a public repository. That makes the data within the study accessible. Secondly, we should be applying metadata. As you're probably aware, data in itself is just ones and zeros and has no intrinsic meaning whatsoever unless you understand A, how to interpret those ones and zeros, and B, what they actually relate to in the real world. And that's where metadata comes in. So for instance, we might have these two columns of metadata, hours of light and location, which provides the much needed context for the data within the study. Okay, that makes it findable by one of these two columns. You could sort and search by these columns, but we can do better. We can use better metadata. So this is a toy example, obviously, but this is metadata that is perhaps taken from a community agreed standard. And in this case, we've gone one better than the previous example because we actually now have ontology terms which unambiguously identify each of these columns of data, and in some cases, the values within those columns as well. So we can take these ontology terms and we can stick them in a search engine, and it can tell us exactly what this column of data represents. And therefore, we can integrate this data with other data, and other platform developers and software engineers and bioinformaticians can write tools based on this metadata, knowing exactly what it means. And then this is what achieves interoperability. And then finally, you put it all together and hopefully data reuse comes out of the other end. And that's the theory, at least. So back to COPO. So as I said at the top, I am the kind of project lead these days for COPO. It's a mature open source metadata and data brokering platform. I think we're in our 10th year now. Originally written to deposit genomic data and metadata to ENA and the INSDC network, because at the time it was thought that this was rather an onerous task that involved an awful lot of wrangling of XML documents, which bioinformaticians and certainly biologists are not particularly keen on doing. Then after Mark Wilkinson's 2016 paper went viral, and the FAIR principles became so well known and widespread, my focus in particular became more on metadata and how this can be used to describe data, essentially. And so these FAIR principles have really guided the philosophy of COPO ever since. So now we aim to be a general deposition service, promoting FAIR metadata in order to improve interoperability. We currently handle genomic reads, assemblies, annotations, images, and other document types. So far, COPO has brokered over 70,000 samples, each of which of those samples has something between, I would say, 40 and maybe getting on for 100 fields. So we're talking between 4 and 5 million data points there. And 45,000 individual data sets to the public repositories. As Sunita said, we are the sample metadata broker for several large consortia, the biggest one, or the most data intensive one, being the Darwin Tree of Life project, for whom we have brokered nearly 35,000 samples. The Darwin Tree of Life project, if you didn't know, is a UK and Ireland-wide mass biocuration project to try and, with the lofty goal of producing reference sequences for all eukaryotic life in the UK and Ireland. We are the broker for the Aquatic Symbiosis Genomics project, who we've brokered over 4,000 samples for, and also the European Reference Genome Atlas project, which is similar to Darwin Tree of Life, but it's Europe-wide and is more kind of infrastructure based. So how do you coordinate a mass curation project across a continent? So I guess one of the main lessons that I learned through this work was how difficult it is to create a consensus. So we had a samples working group for Darwin Tree of Life, and it comprised lots and lots of different people from different disciplines, technicians, bioinformaticians, curators, computer scientists, etc., etc., etc. And they all have a different opinion of what constitutes essential metadata for any given study type. And they're all, valid, and they all need to be taken into account, with the caveat that we can't end up with thousands and thousands of fields for a single sample, because it just becomes quickly unmanageable, and we have to take into account the mental health of the people who have to fill these manifests out. So it's a time-consuming and laborious task. But eventually, after about a year, we had a first draft for the Tree of Life manifest, which is still being refined, and we have a defined schedule for how that happens. And the European Reference Genome Atlas, ERGA, also, it took getting on for a year before that schema was fit for purpose. So this, however, did present somewhat of a problem. So COPO was only adopted sometime after DTOL, Darwin Tree of Life, started, by which time data and metadata collection was well underway. And they had already started collecting certain amounts of certain metadata. So, in fact, had we been there right from the beginning, we could have probably co-opted an existing schema such as Darwin Core or Mixis, probably Darwin Core. It probably would cover maybe 40 or 50% of the metadata that we end up collecting. So that was a lesson learned. However, to turn it into an opportunity, COPO has been developing a translation layer between these different schemas in recent year or two, I would say. So, in fact, there is some power in being able to do this translation. So, as a small subset of an example, we have the Tree of Life some fields from the Tree of Life standard on the left and some fields from the Darwin Core standard on the right. And we can see how these relate between the two. So taxon ID in Tree of Life maps to simply taxon in Darwin Core. Specimen ID maps to organism ID in Darwin Core. Life stage in Tree of Life is life stage in Darwin Core, although spelt differently, and this matters as far as computer systems are concerned because we're case sensitive. So these are two different terms entirely. And GAL, for instance, which stands for Genome Acquisition Laboratory, entry of life, maps to institution ID. So in this way, COPO kind of positions itself as a broker, but also a kind of interlocutor. So in our API, you're able to obtain, no matter under what standard a sample was submitted through COPO, you're able to obtain that metadata, or as much as is possible to map, in one of the other standards. So we kind of go with the philosophy of, rather than trying to impose standards on people, we would like them to be able to communicate with COPO and the wider infrastructure in standards and fields that they feel comfortable and familiar with, and let the work of translating between these different standards, leave that to a system like COPO, because life's too short to be doing that kind of stuff. So back to single cell. So at the Earlham Institute, we have an institute strategic program, part of which is led by Irene, our chair, into cellular genomics, called CellGen, to investigate the impact of genomic and transcriptomic variation at the cellular level in plants and animals. And I'm involved in that. And my tasks essentially are to try and develop and promote community metadata standards for describing these kind of experiments, and developing tools and research data management solutions for handling both that metadata and the data itself, i.e. COPO. So the idea behind this is that we create a beautiful set of well-labeled data and metadata, which helps create tools like cell atlases, for example. So eager not to make the same mistakes that were made with Darwin Tree of Life, I started with the premise that we should be using as many existing standards as possible. So the standard that I have produced, or a schema, I should say, will become a standard when everyone's using it. The schema that I have produced incorporates several existing standards. So MIXIS, which is minimum information about any sequencing experiment, which is a kind of general genomics standard. Darwin Core, which I've already mentioned, which is for biocuration and environmental kind of descriptions. Minsky, which Sylvie mentioned, which is minimum information about single cell experiments, and that was in part done by Irene at EBI and now at Earlham. The Darwin Tree of Life, as I've mentioned, and also FANG, most recently FANG for functional annotation of animal genomes. So what we do is we have this schema, and you choose which of these existing standards should go into your description, depending on which is most appropriate for your study type. We tack on the Minsky fields for the single cell bits, and there's some other fields as well, which I'll talk about in a minute. So we have a website where you can view the schema itself, which I shall show you in a minute. And there's a drop-down list, and you choose which of the existing schemas you want to make the basis for your manifest. Okay. We have different technologies as well. So the single cell RNA-seq bit is the best developed, and we support 10x and SmartSeq. But we also have some emerging standards for VisGen spatial transcriptomics, which I'm talking to various people about at the moment. I'm going to attend a workshop in EBI in a couple of weeks. So if anyone's going to that, I'd love to talk to you about this, which is all about VisGen or spatial image-based spatial transcriptomic data management. So these all go into the single cell schema that I've been developing. And again, there's a drop down and you choose which of those technologies is appropriate to your study, and then I'll show you what it looks like. Okay. So this is all quite early stages of development. I've been working on and off on this for maybe a year now. I published a version 1 of this just before Christmas. It's all on this GitHub, so any and all suggestions are more than welcome. Any feedback. If you're interested at all in this kind of stuff and have strong opinions either way on, fields that should or shouldn't be included, then please get in touch or go to the GitHub and leave issues. I'm more than happy and would love to hear from anybody who is interested. So within that particular project that I just had on the screen, it's quite nicely arranged. So we have a master metadata record, which is an Excel file, and it has all of the fields from all of the standards, where they should appear, on what, under what circumstances should X field appear on the manifest. Things like it has examples, it has descriptions for the fields, it has things to do with validation, such as regular expressions, data type validations, ontology references, all contained in that one file. And then we have some Python code which uses pandas to parse the that master metadata record, and outputs into a variety of formats. The Excel file obviously being the manifest, and then we also have JSON, XML, I believe YAML as well, if you for some reason you want to programmatically ingest the standard. I have been working closely with people at the John Innes Centre, Roslyn, Abraham, all in the UK, and also I co-chair the metadata and infrastructure steering committee on Plant Cell Atlas. So I've been working closely with Ben Cole, who I believe is either talking or chairing the next one of these meetings. So yeah, I'm trying to get buy-in from as wide a range of people and projects as possible, because obviously a standard is only as useful as the people who use it. So I'm trying to be as inclusive as possible and trying to, get as much feedback from the community as I possibly can to actually make this something that's going to be useful for the future. So just to talk about COPO very, very quickly, there is quite a lot of overlap really with what Sylvie showed you in Annotaire. We originally did have kind of web forms like Sylvie showed you, nothing like as nice. But we've eventually sort of found that we'd built almost like a rubbish version of Excel online. And so we thought, well, people are kind of used to using Excel anyway, so why don't we just use Excel? So we ended up eventually casting off our web forms, and all of the ingestion in COPO now is done from Excel files. So we serve XML files. These are downloaded from COPO. So you say, oh, I'm interested in this kind of experiment, and then you press a button, your computer downloads the relevant Excel manifest, fill it out in Excel or whatever you want to use, upload it into COPO, and COPO will then validate so we parse the manifest in pandas, and then validate that metadata. Sorry, sorry, there was a slight interruption. Yeah, we validate the metadata, and as Sylvie showed you in Annotaire, we will report any errors or any warnings. The data can be, we do a lot of just sample metadata, which is then available, we accession to biosamples, and then that's available via our API, which is how a lot of our users use Copo. But we can also broker data as well, in which case the data is uploaded to our staging server, and then the MD5s and file to sample mappings are all done in the manifest. So you just upload the manifest and it does, checks that the MD5s are correct and does the brokering, essentially, transfers the data to ENA, and then you get a project accession. So I just want to quickly show you that website. And then just a couple of example manifests very, very quickly. So if I go to singlecellschemas.org, can move this to the side. That would be good. Right. Okay. So here's singlecellschemas.org. So we can see how it's broken down into different sections. So we have study metadata, sample metadata, dissociation, cell suspension, library prep, sequencing file, and then some other bioinformatics type bits. If we look at the sample metadata, we'll see that this is the Darwin core metadata. So it's incredibly long. And we have, descriptions, examples, where it comes from, regular expressions, etc., etc. If environmental sampling or biocuration is not your thing, you could choose FANG, for instance. And then we have the FANG metadata. So this is stuff about animals, for instance. And then they will all have the Minsky metadata added on top, plus certain fields that, through various conversations and discussions I've had with members of the community, they consider to be important. So I would consider this to be kind of almost the superset of Minsky, because it does have some additional stuff, although it very much takes its cue from Minsky. So the single cell RNA sequencing version is very much the most mature version of this, but we also have draft versions for spatial transcriptomic, image-based, and sequencing. So if you do that, choose, say, tree of life this time, and submit, and then we have a different set of fields. So we have, again, study, sample, file, but now we have imaging protocol. And so these are the files that people I've spoken to who are doing this kind of work feel are important for reproducing their experiments. So that's that. As I said, I sit on the Plant Cell Atlas Metadata and Infrastructure Steering Committee, along with, I co-chair it with Ben Cole. And recently he and Luigi and a few other people have helped me to actually road test this manifest. So the manifest itself looks like this. So these are the tabs along the bottom that you saw here. So yeah, you fill it out. It takes, it's based quite heavily on the Human Cell Atlas manifest, so it should be kind of self-describing. So each field has a description, an example, and then the data kind of starts from this row downwards. And you fill it out as best you can. The fields themselves, they're not all, most of them are optional, I would say. The more you could fill out, the better, and so on and so forth. So anyone who's interested in this, I'd be really pleased if we could talk. I'm going to be at the AG2PI meeting and most of the conference in March. So, if you want to meet offline or even go for lunch, then I'd be more than happy. Is there anything else I want to say? I don't think so. So yeah, thanks to everyone who has helped me with this, and I guess that's it, and I will hand back. Thank you for your attention.
Thank you to both of the speakers. Please may I remind everyone, if you have questions, you can put them on the Q&A or the chat, and we'll be reading them out loud. But first, I will kick off with some questions to start the discussion. So I was wondering,